Hello, my name is Michael Procko, and I am a research scientist at the Peru Lab at the University of Washington. I recently completed my master's in the Wildlife Coexistence Lab at the University of British Columbia, and today I will be talking about one of the chapters of this master's thesis, which was recently published. This is titled Human Impacts on Mammals in and Around a Protected Area Before, During, and After COVID-19 Lockdowns. So at any point during this presentation, you might see the QR code. That'll take you right to that publication if you're interested in reading more about it. So diving into what I'll be talking about today, I'm going to provide a brief introduction on recreation, protected areas, and wildlife more generally. Then I'll get into more of the specifics of my study area and my study design. I'll talk a bit about this general summary of what we caught on our camera trap survey, and then I will move into two different types of analysis, each at their own temporal scales, which kind of help us investigate the impacts of recreation on wildlife in this system. And then I'll wrap it up with some big takeaways and conclusions. So protected areas are a pretty common strategy for mitigating the impacts of human activity on wildlife. However, recently their effectiveness in conserving biodiversity or conserving wildlife more generally is pretty commonly debated. Now, 95% of protected areas in North America are publicly accessible, which leads to quite a bit of recreation taking place in these landscapes. And more in the literature, we're seeing that recreation may actually reduce protected area effectiveness by imposing negative effects on the wildlife inhabiting these spaces. Impacts of recreation on wildlife might include habitat loss for some particularly sensitive species. It could also include changes in behavior, such as species becoming more nocturnal or crepuscular in areas of higher human activity. At a physiological level, recreation may also increase stress or anxiety and reduce reproductive success. And cumulatively, over longer periods of time, all of these different impacts may result in trophic structure shifts or just changes in community composition in these spaces where recreation is commonplace. So to observe whether recreation was impacting wildlife in my particular study area, I was working in a provincial park called Golden Ears Provincial Park, which is about 40 kilometers east of Vancouver, British Columbia. Now, Golden Ears is a massive recreation hotspot in British Columbia and recently hosted the #1 most annual campers and the #2 most annual day users. However, the park does not permit off-road recreation, so much of the human use of this park is limited to these quiet activities such as mountain biking, horseback riding, and hiking. Nestled to the southwest corner of Golden Ears is Malcolm Knapp Research Forest. This is a university-managed research forest owned and operated by the University of British Columbia and is primarily used for research and education activities that are associated with the university. There's a wider sprawl of roads throughout this landscape, which are primarily used for research activities, but can also include harvesting, thinning, and replanting, which is just something that the university does to fund the maintenance of this forest. So this landscape represents a pretty interesting mosaic of human activities, ranging from these pretty tremendous quiet use recreation activities to these louder working activities that are kind of isolated in Malcolm Knapp. Now, throughout this landscape, we deployed 58 camera traps. 37 of these were in Golden Ears, with 20 of those being along trails and 17 being off trail. And then 21 cameras were also deployed in Malcolm Knapp, with 13 of these being along trails or these restricted access roads, and 8 of those being off trail. Now, we started setting these cameras up in March of 2019, and my final data collection took place in September of 2020, which includes this really interesting period of time from April to June of 2020, where both landscapes were closed to the public due to the COVID-19 pandemic. So a summary of what we caught on our cameras, we detected over 1 million images from about 24,000 camera trap days, where one day that one camera is active is considered one camera trap day. In our images, we detected over 108,000 hiker detection events, over 1,000 vehicle events, over 600 mountain biking events, and over 900 horseback riding events. So quite a lot of human use taking place in this landscape. In addition to the human uses, we detected 19 mammal species, but for the purposes of this project, I am focused on the 6 most commonly detected species, which were black bears, mountain lions or cougars, black-tailed deer, snowshoe hares, bobcats, and coyotes. Now, with respect to the specific questions we had about these species, we were interested in two main hypotheses. The first is the predator shield or human shield hypothesis, which ultimately states that predators often avoid humans, which allows prey to associate with these spaces using humans as a sort of shield from predation. And we're also interested in testing a similar hypothesis called the mesopredator release hypothesis, which ultimately states that predators do tend to avoid these spaces of higher human activity, and therefore mesopredators could be released from top-down pressures, so predation or competition, in these spaces where humans are more prevalent. So as I mentioned, I tested these hypotheses at two different temporal scales. I'll first talk about the weekly scale. So for each of our 6 focal species, we built one Bayesian mixed effects regression model, which ultimately just regresses the probability of weekly habitat use against weekly recreational pressures, as well as other variables expected to influence habitat use. Now, the response variable in these models is just a binary indication of whether a species was detected in a station week, where 1 week that one camera is active is 1 station week. The variables that we included in each of these 6 models included the weekly hiker detection rate, the weekly mounted recreationist detection rate, and the weekly motorized vehicle detection rate. And these are really our human activity variables that we're most concerned with when it comes to our hypotheses. Now, I should note that mounted recreationists includes both horseback riders and mountain bikers. These had to be clumped together because they were highly collinear, being restricted to kind of the same locations. But in addition to our human use variables, we also included a number of environmental variables just to control for any sort of variation that may be caused by the environment. These could be something like crown closure, NDVI, or the distance from the camera to the urban wildland boundary. We also included camera set variables, as we know that the differences in how the cameras are set can often result in differences in our conclusions. We did try to standardize this to the best of our ability, but just to control for any sort of variation that we could not control for in our standardization process, we included variables such as the camera height and the distance from the camera to the anticipated path of the target. But finally, we also included camera station as a random effect, which really just accounts for any non-independence among weeks sampled at the same station, as well as any temporal autocorrelation. So we assessed the strength of evidence for a variable impacting species habitat use as those whose 95% credible intervals did not include 0. So first and foremost, the human detection variables that we identified as having a strong impact on species habitat use were hikers for bobcats and motorized vehicles for black bears. Now, bobcats have been noted as potentially sensitive to recreation in other contexts, so we provide further support for previous literature that has identified these trends. However, black bears, this finding was particularly interesting as this species is well known to select for regenerating stands following logging events because they often contain berries. So we provide a pretty novel finding that they may be negatively impacted by the vehicles used in forest operations, despite their tendency to inhabit these spaces once vehicles have left. Additionally, an interesting but unexpected finding was that all mesopredator and prey species were more likely to be detected at areas near the urban wildland boundary. So while we did not necessarily find support for the predator shield or mesopredator release hypotheses looking at just human detections, this may provide a sort of landscape scale support wherein we might see these species using the residential or agricultural areas adjacent to the park as a shield from predation pressures. And then finally, there were a number of other environmental variables and camera set variables that were important, which further emphasized the importance of us controlling for these alternative sources of variation in our data. So moving into the second scale that we tested, we similarly built Bayesian regression model for each of the same species. However, in these models, they were much more simple, where we simply regressed the detection rate of each of these species either before, during, or after the closure against a factor variable which represented that time period. And so our detection rate is just our number of detections in that period over the number of days the camera was active in that period. To illustrate the periods of time we used for pre-closure, closure, and post-closure, I've constructed this plot of human detections across our entire study area throughout time. And you can see the blue hashed lines, or the space between the blue hashed lines in the summer of 2019. That's our pre-closure time period, whereas the blue hashed lines, or the space between these blue hashed lines in the summer of 2020, is our post-closure period. And perhaps a bit obvious due to the tremendous drop in human activity, our space between the red dashed lines is the closure period. Once we constructed our models, we found only 2 species showed changes in their detection rates throughout these times, with cougars being more commonly detected during the closure period than before or after, and black-tailed deer being detected at similar rates before and during, but more common once the park reopened. So this provides a bit more support for the predator shield hypothesis, and we see this tendency for mountain lions to maybe not inhabit these spaces of human activity when there are a ton of humans, and then as the humans were removed, mountain lions were more capable of moving in. Another interesting point in this is that following the reopening of the park, a trail limit system or a day use pass system was implemented, which kind of implies that black-tailed deer may not be able to tolerate these heavy recreational pressures that we saw before the closure, but with this new trail restriction system, we might see more black-tailed deer. So our big conclusions coming out of this study: we know that recreation may indeed impact wildlife, but this impact, or these impacts, are often species-dependent, disturbance-dependent, or scale-dependent. And furthermore, that the context of the study area is incredibly important. We see this especially with our distance to the urban wildland boundary being a pretty strong predictor of a lot of these species. We also make a big mention that camera traps were incredibly effective at collecting data even when we were no longer allowed to be in this landscape. We were not exempt for the first bit of this closure period. So it's very fortunate that our cameras were capable of collecting data even while we were not allowed in this area. We also want to make a big mention that protected area management requires really careful consideration of balancing both wildlife and recreation, and furthermore that thresholds in human activity where these species may be able to tolerate certain smaller levels of human activity, but not these massive levels. Those are kind of the key to translating our science into policy, and identifying those is really crucial for effective protected area or wildlife management. So with that said, thank you so much for listening. If you have any questions, please feel free to reach out. And like I mentioned previously, if you want to read more about this paper, you can scan the QR code right there. Thank you.